Welcome to this podcast series, which is brought to you by the Young Epilepsy Section of the British Branch of the International League Against Epilepsy.
Thanks everyone for being here today. Dr. Gareth Morris, a lecturer in neuroscience at the University of Manchester and ERI Emerging Leader Fellow at UCL, and myself, Dr. Faye MacLeod, an ERI Emerging Leader Fellow at Newcastle University, are delighted to interview Mr. Ashan, Jayasekera and Professor Mark Cunningham on the use of human brain tissue for epilepsy research. Mr. Ashan Jayasekera is a locum consultant neurosurgeon at St George's Hospital in London and former ERI Fellow. And Professor Mark Cunningham is from Trinity College Dublin, and his research focuses on understanding the basis of neurological and psychiatric disease at the level of the neuronal circuit, microcircuit. So, Gareth, are you ready to start the first round of questions?
Yeah, sure. I can kick off. So, thanks, Faye, for the introductions. It's really exciting to have this podcast together. Thanks to Ashan and Mark for joining us and sharing their wisdom with us. So I suppose the first question really, could you both just tell us a little bit about what experiences you have of working with human brain tissue for your epilepsy research? You know, how does this work? What sort of stuff have you done with this tissue? Maybe Mark, if you want to kick us off, and then we'll pass over to Ashan to give us his side of the story.
Yeah, sure. And thanks again to both yourself, Gareth, and Faye for this invitation to speak to you both this morning. So my journey with using human brain tissue really started from the perspective of my background is I'm a preclinical researcher and have a track record of using a lot of preclinical animal models, but got to a point in my career where I was trying to explore more sort of humanized models. And of course, the ability to use human tissue presents a fantastic resource to do that, and particularly for an electrophysiologist, so somebody that's trying to study how neurons and synapses and circuits organize themselves to generate organized electrical activity. And of course, epilepsy is a very interesting disease because from a clinical point of view, we use a neurophysiological tool to diagnose it and to monitor it, which is the EEG. So initially, my interest was to really sort of start looking at human tissue that we could collect from classical epilepsy surgery. So these would be cases like temporal lobe epilepsy, and to try and look at those samples and see whether we could understand something about how those circuits generate organized electrical activity. And indeed, in our first publication, we demonstrated that these samples could actually generate spontaneous epileptic discharges. So we didn't have to do anything sort of to modify that material. The tissue would actually start to generate these spontaneous interictal spikes, which is what you see in the human condition. And we did a number of studies there, for example, where we're looking at the contribution of electrical connections, gap junctions to that activity. And then my interest started to evolve, and this is perhaps a nice segue into Ashan, when I started to get more interested in brain tumor-related epilepsy. And we discovered that we could, for example, get tissue that was coming from patients who primarily have a tumor within the brain. That could be either a low-grade or a high-grade tumor, but also have an associated epilepsy phenotype. And again, that sort of posed a very interesting set of questions in terms of how the tumor is interacting with the surrounding brain cells to generate a seizure activity and a focus around this region, which is called the peritumoral region.
Yeah, that's perfect. Thanks so much, Mark, and it's really interesting research. So Ashan, I suppose you're on the other side of the equation here. You're in the surgical theatre, you are the neurosurgeon. So what is your experience of working with human brain tissue in your sort of involvement in the research side? Obviously, Mark's in the lab studying the tissue. What's kind of your role in the research, if you like?
Thank you very much, Faye and Gareth, for the invitation. So I know Mark very well. Mark is the person who got me into research in the first place. So I was a neurosurgical trainee hanging around in theatres, and this man would turn up with a bucket and oxygen cylinder, and we used to call him the brain snatcher. And when the patients were having their tumors taken out, the surgeon would usually, as part of that resection, gift a bit of tissue to Mark, who then take it down to lab and sort of have a look. So I followed him down one day, and I saw him, slice the tissue up, put it like in a dish, and put some electrodes in. And obviously, it was fascinating seeing what he could do. Like, he could detect these spontaneous discharges. He could then push the tissue into a seizure. And from my point of view, the thing that I've always been interested in for patients with brain tumors is, as Mark said, a lot of them have an epilepsy phenotype, but a lot of the treatments that we offer patients aren't there to offer them seizure freedom. So my interest was really on, what could you do for these patients that would help you remove the tumor, but also render them seizure free. Now, anybody who works in the field of epilepsy knows that for patients with epilepsy, one of the things you'd often try to do is you try to characterize the seizure-generating brain using a combination of scans, but also using implanted electrodes. So electrodes that you put on the surface of the brain or into the brain to try and characterize which bits of the brain are generating a seizure. The problem with that is it's quite an invasive investigation. That carries risks. So we know that about 23% of patients who have these kind of procedures will have a complication. And of those patients who have a complication, 9% will need an operation to remedy that complication. So my question was, is there a non-invasive way of mapping those bits of brain around a patient's tumor that are responsible for their seizures? And I was interested in using a technique called MR spectroscopy. So everybody's familiar with standard MRI scans. They produce a nice anatomical picture of a patient's brain. An MR spectrogram looks at the chemical composition, and I was particularly interested in a molecule called glutamate, a neurotransmitter, to see if that could identify regions around a tumor that were responsible for patient seizures. Now, if you're going to use this new technique and compare it to the technique that's usually used, these invasive implanted electrodes, what you would typically do is you'd run a study where you'd obtain some MR spectrograms around a tumor, and then you'd implant electrodes in and around the brain tumor to try and identify if the two were correlated. But invasive electrogram recording is not part of a glioma patient's workup. So we needed some kind of bridging technique, and that's really where I thought that the stuff that Mark was doing is very exciting. So what we did instead with these patients was we did some MR spectroscopy recordings around the tumor, and then as the tumor was being resected in that same region, we obtained some of the tissue and then used the techniques that Mark's been developing in his lab to see if we could see any spontaneous activity in the tissue. And that way we could study an association with the MR spectroscopy and the seizure-generating potential of tissue around a patient's brain tumor. So that was the kind of my journey into human tissue work.
And if I might just come back in, I mean, one of the reasons why this glutamate signal is so interesting is, our brains use glutamate as a normal neurotransmitter for neurons to communicate with one another. But what seems to be occurring in brain tumor-related epilepsy is that the tumor actually hijacks the glutamate signal for its own ends. So it uses glutamate, it produces lots of glutamate, and glutamate actually gets to very high levels where it becomes excitotoxic. And what that means then is that that excitotoxic glutamate is actually killing normal brain cells to create more space for the tumor to invade. But of course, glutamate is also then making the brain more excitable. So it's a very interesting neurotransmitter. And Ashan's work is very elegant because it actually gives us a useful biomarker that could be then used to interrogate, for example, novel therapies that might aim to target that excessive glutamate and to obviously see whether they can reduce the glutamate and dampen down the seizure activity in those patients with brain tumor-related epilepsy.
Okay, thank you very much. That's a really interesting overview of the different types of research that you guys have got into, especially around in terms of tumor and resecting the tumors and being able to understand, how the source of potentially where the seizure is coming from and the mechanisms behind that in terms of treatment. But I just wanted to kind of take it back in terms of to you, Ashan, more beyond sort of the tumor-related epilepsies. What can you tell us more about the kind of the surgical procedures and why they're needed for other types of epilepsies, and what the outcomes could be, and how maybe the research that could be done maybe with ex vivo human tissues, or just in general with the type of surgeries that are going on with at the clinical side?
Yeah, of course. So of the patients who present with focal epilepsy, so that's epilepsy that manifests in a particular region of the brain, as opposed to kind of generalized tonic-clonic seizures that we're familiar with. About 60% of those patients will have their seizures controlled. But the flip side of that is 40% won't have the seizures controlled despite being on 2 or more anticonvulsant drugs. So you have to look beyond anticonvulsant drugs for other treatment options. Now, people have obviously talked about the ketogenic diet, but one possible treatment option is surgery. And surgery for epilepsy really comes in sort of 3 forms. You get resective operations, where you've got a clear lesion that's generating seizures that you attempt to resect. You get palliative procedures where you can't resect the epileptogenic focus, but you can try and stop the spread of seizures. And the classic example of that is a corpus callosotomy, where you divide the white matter tracts that connect both hemispheres. And then you can get other stimulation procedures, things like vagal nerve stimulation, deep brain stimulation, that won't render a patient seizure free, but they can certainly try to control or reduce the number of seizures. Now, the simplest scenario is somebody presents with a seizure, has a scan, and there's a clear lesion on the scan. There might be a tumor, a cavernoma, hippocampal sclerosis. And the seizure semiology, the kind of seizures they have, match the area within the brain that the lesion is in. And the scalp EEG recordings they have, these electrodes that get put on the scalp, suggest that the seizures are actually originating from that region of the brain. So that's a simplest scenario and in those patients, they will respond quite well to having the seizure focus removed. It becomes a bit more complicated if they don't have a lesion on their scan. There's nothing on an MRI scan. If they have a lesion on the scan, but it doesn't quite match their seizure semiology, or that the seizure-generating region of brain is quite close to eloquent cortex, so it's a near bit of the brain that's responsible for speech, for patients' motor function. In those patients, you need to have a more considered approach. So you have to establish where the seizure-generating focus is. And you can do that, as I mentioned earlier, with invasive electrodes. You can do it with other kinds of scans. And if you can identify a seizure-generating focus using that, then again, those patients you can offer surgery. Now, there will be some patients where, despite those investigations, it's not possible. And those are kinds of patients who might have a palliative procedure like a corpus callosotomy, who might go and have a stimulation procedure. The success rates for a patient has a lesion are quite high. So, for example, the classic example of epilepsy surgery is a condition called hippocampal sclerosis. And the initial seizure freedom rates of being entirely free of seizures within the first year is around 75 to 80%. Okay. Outside the temporal lobe, the seizure freedom rate drops. But for patients who have disease in the frontal lobe or parietal lobe, for those patients who have a resective procedure, the seizure freedom rate is about 50 to 60%. So epilepsy surgery does have a role to play. And I think one of the interesting things about patients who are having surgery for epilepsy or tumors is there's a whole data set of clinical information, pathological information, radiological information. I think for me as a clinician, that's really the exciting thing about using human tissue, is that you can take this human tissue, perform some electrophysiological recordings, pharmacological experiments, but then you can relate that all back to the patient's clinical history, how long they've had seizures for, their age, their sex, the underlying pathology, what their scan shows. And that was the thing that really excited me about using human tissue as a clinician.
Yeah, no, I absolutely think that's been able to map back, I think, to the kind of clinical phenotype, or the kind of whether they've got seizures, what drugs they're on, for example, will really help to be able to correlate the findings that you have both from in the lab and to the patient in terms of a kind of two-way system. Gareth, you were going to now focus on the specific overview of the type of findings that we can have. Is that correct?
Yeah, I mean, what I might do is just jump back to the previous question slightly, and I'm not sure, maybe Mark's the person to answer this question, but so Mark, this is something you and I have talked about in the past. It's quite important, the manner in which the surgeon removes the tissue from the brain. That, we've talked about needing to resect as a block to sort of help the follow-up procedure. I thought just for people who haven't worked with this kind of tissue, could you just very briefly touch upon the sort of way in which the tissue is ideally prepared from, talk us briefly through how does it come from a person's brain into the lab?
Yeah, no. I mean, the core part of this really is and this is critical for this endeavor as a whole, is really a fantastic collaboration and interaction between scientists like myself and the neurosurgeons who are removing the tissue. And if you don't have that relationship, then it's difficult really to get the types of recordings that you want to do. So I have to admit, the first time I saw neurosurgery, I was actually quite horrified, because as Ashan, I'm sure will say, there's a lot of tools and implements that are used in neurosurgery. I mean, for example, they use diathermy because the critical thing for a neurosurgeon is to actually minimize the loss of blood. So they have these forceps where they can diatherm the tissue. And actually what they normally do when they're removing tissue is they use the diathermy to sort of to cut around the tissue and essentially remove the tissue. Now, if you want to study live tissue, that's not great because the tissue is going to be damaged. So what normally happens is, in our experience, is that we would arrive in theatres usually in a timely manner just before the surgeons are about to remove the tissue. And when ready, the surgeon will essentially use a blade, a microblade, to essentially cut out a small cube of tissue that's part of the lesion that's going to be removed anyway as part of the clinical procedure. So this block of tissue can be anywhere in the region of 1 centimeter cubed or a couple of centimeter cubed, would normally be going to clinical waste, and we're standing there essentially with this small container of what's called a artificial cerebrospinal fluid. So normally our brain sits in this fluid, and we can make this solution up in the lab. It essentially just consists of a combination of salts made up in water. Now, what's slightly different about this artificial cerebrospinal fluid in terms of collection is that it's kind of slightly frozen, so it looks like a bit like a slush puppy. And that is because we want to essentially minimize any sort of damage to the tissue during collection, and also before we can get it to a specialized piece of apparatus which is called a vibratome. And what that vibratome then allows us to do is cut very thin sections of tissue. What's also occurring is that the little container of human CSF is being bubbled with carbogen, 95% oxygen, 5% CO2, because again, we want to keep the tissue in a healthy environment. So what we're trying to do is essentially fool this bit of tissue into thinking it's never left the whole brain. So we're supplying it with all the goodies and nutrients that it would get if it was still in the brain. And when we get it to the vibratome, then what we're doing is we need to cut it into slices. So for in vitro electrophysiology, brain slice electrophysiology, what you're trying to do there is you're trying to prepare slices that are thin enough that you can continue to keep the tissue alive, but thick enough that you still have the body of the neuronal microcircuit. So the neurons and the synapses and all of the other cells, astrocytes, microglia, that allow you to then study the networks within the brain. So generally we cut these slices at about sort of just under 0.5 of a millimeter, about sort of 400 to 450 microns thick. And then what that means is that you can keep those slices alive for, in many cases, 24 hours. And there's also now techniques that are being deployed using organotypic approaches, which keep the tissue alive even longer. So instead of a day or 2 days, you can keep the tissue alive for weeks, and that gives you unparalleled opportunity in terms of then looking at, for example, expressing viruses in there, looking at mRNA approaches. So you can really start to explore very novel therapeutic avenues. But again, it really comes back to that very first step, which is the critical removal stage of the tissue. And again, unless you have a very good understanding with the neurosurgeon, in many cases, you know, if you don't get good tissue, then all of that other exciting stuff is doomed to fail.
That's great. Thanks so much, Mark. A really sort of clear overview of how this sort of stuff all actually works. So I went slightly off script there. We'll come back to script, which brings us to you again, Mark. Maybe you could just tell us quickly. So you mentioned sort of research into glutamate and sort of with peritumoral tissue. Are there any other notable findings that you've made using human tissue in your research?
I mean, I think I go back to the first one, which was the contribution of gap junctions. When we think about how neurons communicate with one another, we often think of the kind of classical neurochemical processes using glutamate, the excitatory neurotransmitter, or GABA, the inhibitory neurotransmitter. But for a long time, we've known that there are other forms of connections between neurons, and these are gap junctions. So these are specialized proteins which actually allow direct electrical connectivity between neurons. And particularly in a disease like epilepsy, where you get very rapid synchronization of electrical activity across the brain, it's long been thought that these gap junctions might play a role. So we were able to demonstrate that indeed they do, and they're very important for producing particular types of patterns of electrical activity, which are called high frequency oscillations, which are known to be important from a clinical point of view in terms of defining regions of the brain that are involved in the epileptogenic process. We're sort of at the minute trying to follow up on a number of those observations, but we're also again trying to follow up on the whole story around glutamate. And for example, we're trying to look at some of the specific receptor subtypes that glutamate acts on, that are also known to be key to the pathology of epilepsy, but also targeted by conventional anti-seizure medications. So for example, one type of receptor is the AMPA receptor. And what's very interesting about the AMPA receptor is, again, it's known to be located on synapses to facilitate the glutamate causing excitation. But what's very interesting is that there's recent evidence to suggest that the glioma cells are actually now making direct synaptic connections with normal brain cells. So for a long time, we kind of thought, well, you've got the glioma and it's quite, it's a separate entity to the surrounding normal brain cells. But there's a lot of evidence now that is showing that there is actually intimate relations, both through these AMPA-specific neuroglioma synapses, but also through gap junctions as well. So it's a very complex biology, but I think the more we understand it, then the better chances we have of actually going in and coming up with better treatments for brain tumor-related epilepsy.
Thanks for that. That's actually really interesting to be able to see the different types of findings that you guys have been, what you've been doing, and also the treatment options of that, and actually the exploratory type of research that gets done within this. I have in terms of so obviously what we're working with here is it's the human tissue. We're working with, the real deal with what we're doing, but a lot of the research so far over many years has been done using animal models of different types of epilepsy. And I know that we can do similar techniques in terms of looking at the functional activity within acute slices in rodents. Do you think, and this is to both, Mark and Ashan, do you think this is still important that we actually compare and contrast the differences between both the acute and in vivo animal models with the human tissue work as well, and also the findings that we have, are they comparable? between the two, and how can we work with both the animal models and the human tissue work?
Shall I start?
Ashan? Yeah, you go first, Ashan.
So I think, as Mark would attest to, one of the frustrating things with all this preclinical work, there's been a failure really to translate a lot of the findings from bench to bedside. And even with the kind of glutamate in glioma story, there was a lot of preclinical stuff, but kind of died, and there hasn't really been much published since a lot of the work done by Sontheimer. So it's really important to kind of confirm whatever you find, I think, in a preclinical model with a clinical model. I think that's one of the kind of key things that I would sort of say. If you definitely need this kind of bench side work with animal models, because the stuff that Mark and I did with glutamate and glioma would really not have come about if you hadn't been tested in a preclinical model. But then you have to confirm these findings in human tissue, I think.
Yeah, I would completely support that. I'm actually at a human neuron microcircuit meeting here in Amsterdam at the minute, and one of the speakers this morning, it was different disease, Alzheimer's disease, came up with this phrase that we've cured Alzheimer's disease hundreds of times over in mice, but we haven't done it yet in humans. I do think there's room for both, and as Ashan said, I think it's important to have that aspect of translation between animal models and human models. I mean, I think there's also another point here, which is what I call comparative neurophysiology. I actually think it's very interesting to study from an evolutionary point of view how the difference between microcircuits in mouse and rats and humans, how they've evolved, and the actual divergence that has occurred. Because again, I think that tells us potentially some very interesting things then about how certain diseases arise, and perhaps tells us or gives us at least clues about how we can tackle human disease. And again, then obviously try and back translate that into preclinical animal models to actually make them more realistic from a human point of view.
Yeah, thanks both. That's some really interesting insights. And the next question kind of follows on from what you've just said there, Mark. And that is, how translatable do you think these sort of human brain slice models actually are? Obviously, they're a real piece of human brain, which is fantastic, but it's still a sort of confined piece of tissue in a dish. So what are your sort of thoughts on translatability? Maybe we start with Mark and pass it to Ashan.
I mean, I think I come back to my point about biomarkers. I mean, the type of work that we do, which is using electrophysiology, and particularly electrophysiology that tries to interrogate how the circuit operates. So, for example, using local field potential recordings, that's very translatable, because it is the local field potential which actually is what generates the EEG signal. So I think if you're interested in how circuits behave, then looking at, for example, organized electrical activity at a network level is very, very translatable. I mean, I think the important question is obviously thinking about, for example, efficacy. So the real advantage that human work has, particularly, for example, from a pharmacological point of view, which is potentially very translatable, is the ability to interrogate therapeutic options. That could be pharmacological, it could be these newer modalities like gene therapy, for example. And I suppose to then give clinicians the confidence that the method, the therapeutic methodology that one has used has been shown to be successful in primary human disease tissue, and I think that gives then confidence to clinicians who are then maybe, for example, designing clinical trials, that at least they have some relevant readout that has been done in the human sphere, albeit at an in vitro level, of course, and that's an important caveat as well.
So Ashan, maybe I can bring you in to sort of share your opinions on that from a more clinical viewpoint. If you're involved in research, is it reassuring to you to see this human brain tissue being used? Or, would you be happy with animal models? Is the human brain tissue more translatable in your opinion?
Yeah, I think so. I mean, obviously, the difference between directly studying a patient with epilepsy versus modeling it in an animal. And certainly, if you look at a lot of the initial studies done, a lot of the things you can see in the acute slice work. So, for example, these spontaneous epileptiform events that Mark was seeing in his lab, they are very strongly associated with the kind of electrophysiological events you can get with direct recordings, so in vivo recordings in the theater. So I think what you're seeing is, part of the epileptogenic process, and you're directly studying it. And if you look at the changes within the tissue themselves, there are lots of pro-epileptogenic changes which are clearly biologically plausible in terms of generating epilepsy. So in and around tumors, for example, you see a loss of inhibitory synapses. Electrophysiologically, you can see evidence of chloride loading, these depolarizing GABAergic phenomenon. So I think so. It's very much is the case. I think for a clinician like me, it's a nice bridging technique, as I mentioned earlier, to try and study using other modalities like MR spectroscopy to hunt for seizure-generating brain around tumors. It's also a nice, I think it's a nice technique to test out new anticonvulsant drugs, generate a seizure in a lab, actually promote a seizure in the lab by changing the ionic concentration using proconvulsant drugs, and then test the efficacy of an anticonvulsant drug before testing it, in a phase 0 trial in a patient, rather than directly from an animal model to a patient.
Okay, well, I think that leads us on quite nicely to our kind of final part, which is where, and this again will be to both of you, is like, where do you see the future of this field going? And I think one of those could be, for example, as a, could it be as a platform to test, for example, novel drugs, for example, from different types of kind of tissue samples that are coming from different types of seizures, for example, or different patients with different types of epilepsy, or being able to generate that in the lab, for example. Where else? I think you've also touched upon this, Mark, as well, with the organotypic slices, which is a new kind of, in the last sort of year or two, there's been a lot of labs around the UK and also the world who have been trying to promote this, as I try and promote the longevity of the tissue, to use it for maybe modeling genetic epilepsies, for example, or even just trying to different mitochondrial diseases associated with epilepsy, for example. Where do you think this? Do you think that's the main kind of point for this tissue going forward?
Yeah, I mean, I think obviously this all started from an epilepsy focused perspective because of the nature of these patients, but certainly what I'm seeing is that people are looking at this technique now, and particularly the organotypics, and really starting to think about whether it can be used to study other types of disease. For example, Alzheimer's disease, mitochondrial disease, other neurodegenerative disorders. So I think the organotypics, again, give real advantages in terms of the longevity and looking at novel methodologies to study these diseases. But they also, again, I go back to this point about novel therapeutic options in terms of gene therapy approaches. The organotypics really give the opportunity to do that.
Brilliant. And what about you, Ashan? Where do you think the future of this field is going?
So I'm a simple neurosurgeon, so my view on it is less highbrow than Mark's. But what I'd like to see is that anybody having an operation to resect a tumor or having epilepsy surgery, they should be considered to be part of a study that samples human tissue. And I guess that would be what I would want to see, that all these patients have the opportunity to be part of these studies, but also that databases are kept, so we have a repository of information where the scientists collect any electrophysiological measurements, any pharmacological tests, and a researcher who doesn't have access to human tissue can access that repository and look at the clinical information, look at the pathological information, look at the radiology. Because really, if you go to, because that's what you want lots of eyes on the problem, lots of researchers from other units being able to access this information, who may not have access to human tissue, but have important questions they want to answer.
Yeah, and that kind of work is kind of underway. So I'm part of an MRC UK network, which is trying to build this kind of capacity. Because again, not everywhere can do these types of studies. It can be technically challenging. You have to have, be close to a site where there is neurosurgery taking place. So, and also there's, this question of the kind of speed of this type of work. Obviously, it very much depends what cases present, and just the bottleneck in terms of having the resources to do the types of reporting. So that kind of network approach can really work to maximize the amount of data that's being collected, but also shared as well.
Okay, brilliant. I think that probably is a good ending point for the moment in terms of where we think the future of this field can go. I'll just end by saying thank you again, both to Mark and Ashan, for taking the time today to talk to us about their research and their thoughts on the field of using human tissue in epilepsy research. And on behalf of the British ILAES committee, we look forward to obviously seeing what research and kind of where things are heading from here. So thank you both again, and watch this space.